Chickpea is one of the important pulses we eat. India is the major producer of chickpea. In India, it is cultivated under both irrigated and rain-fed farmlands. Out of total chickpea cultivation land, 85% are rain-fed, where drought and fungal disease cause yield reduction. Fungal diseases such as collar rot, sclerotinia stem rot and fusarium wilt occur and aggravate irrespective of soil moisture status. Exceptionally, dry root rot disease, which is viewable as dried plants, is aggravated only under low soil moisture conditions. Dry root rot is one of the economically important diseases caused by the fungal pathogen Rhizoctonia bataticola and aggravated by drought stress. Dry root rot diseased plants are devoid of lateral roots, and contain microsclerotia in the pith. Rhizoctonia bataticola produces microsclerotium and mycelium. Microsclerotium is a dormant structure, wherein the cells are cemented together in a heavy melanin-pigmented gelatinous matrix. Mycelium is a vegetative structure. Under laboratory conditions, the fungus can be maintained in potato dextrose agar media in Petri plates. The plant root is the main target of Rhizoctonia bataticola as it is a soil-borne pathogen. In the display shown, the black dots present on the root are microsclerotia. On the left side, transverse section of the root is shown. From periphery to center, the root has epidermis, cortex, endodermis, pericycle, phloem, xylem, and pith. On the right side, peripheral structure of the root is shown. The plant releases root exudates. These exudates attract fungal pathogen. Upon sensing the exudates, microsclerotia attach to the root epidermis and germinates and germ tube emerges, which infects the root epidermal cells. Fungus secretes cell wall degrading enzymes and toxins, which causes tissue maceration, which results in local necrosis. Initial necrosis marks the success of infection. After the establishment of infection, the fungus colonizes the cortex region. At the same time, hyphal branching event occurs, which is the hallmark of initiation of severe infection. The branched mycelia forms microsclerotia and further extends the infection. Then, colonization of the entire cortex and epidermal surface and formation of more microsclerotia occur. Furthermore, colonization of the vascular region starts. Finally, fungus colonizes the xylem and pith region and causes root rot with formation of more microsclerotia. The colonized plant roots eventually rot, which results in the death of the whole plant. In the display shown, plants growing under sufficient amount of water in the presence of fungus has less infection and healthy lateral roots. Plants growing under water deficit condition has more infection and lack lateral roots. In the display shown, when the plant is growing under well-watered condition, the infection does not colonize beyond cortex region. Also, later root number increases under well-watered condition. However, when the plant is under drought stress, the colonization extends from cortex to vascular region, where it destroys the xylem and pith and kills the plant. The control, uninfected root is lack of microsclerotia and root cells are turgid. The infected plant root under well-watered condition, a few numbers of microsclerotia are present. In contrast, infected plant root under drought stress has a greater number of microsclerotia as well as dried rotten root cells. In conclusion, dry root rot is an emerging threat to the chickpea cultivation in India. Several chickpea growing regions in India receive less rainfall and hence face a water deficit crisis, which is leading to increased incidence of dry root rot in the field. Our lab has established that dry root rot incidence and severity in chickpea is positively correlated with drought stress. We found that under a well-watered condition, plants restrict the fungal infection and produce new lateral roots, which helps in foraging nutrients and water. Under drought stress, plant is predisposed to fungal infection. Also, drought stress aggravates the disease severity. Our lab is uncovering the unknown plant response mechanism to dry root rot pathogen in chickpea. Using this knowledge, we are trying to identify the genetic resistance to dry root rot from a genetic pool. In addition, biotechnology tools are used to develop an improved variety with resistance to dry root rot.